The mitotic phase of the cell cycle does have two parts to it. The first part, or what we tend to think of as the first part, is mitosis or karyokinesis, and then the second part is going to be cytokinesis. In mitosis, it is really division of the nucleus and the stuff that's in the nucleus. But when we get to cytokinesis, cytokinesis is division of the rest of the cell. And a lot of times we think of this as division of the cytoplasm, but it's the cytoplasm and all of the stuff that's contained within the cytoplasm. And so the overall mitotic phase, or the M phase, really isn't complete until we do have both of these processes take place. So let's talk a little more about cytokinesis and the things that are going to take place during that phase. The process of cytokinesis itself usually starts or begins during anaphase. And this is why when we look at a cell that is in anaphase, usually we're seeing an elongated cell. So if that happens to be an animal cell, since they tend to be a little more spherical, we start to see more of an oblong cell. If we're talking about a plant cell, which tends to be a little boxier, we're going to see it become more of a rectangle rather than that little square shape. So just the lengthening of the cell itself indicates to us that we are beginning the cytokinesis process. Now, the way that the cell is actually going to lengthen is that we will have special microtubules involved, which are called polar microtubules. The polar microtubules, they do come out of the centrosomes. So if we were to draw some centrosomes here, recall that the centrosomes are going to be at opposite sides of the cell, and we would certainly have spindles that come out of them because they are responsible for making the spindles. Some of the spindles do attach to chromosomes that are located down the center of the cell, but there's also going to be some microtubules that do not attach to chromosomes. So these are the polar microtubules. These do not attach to chromosomes. And instead, they will lengthen the cell or help to really kind of stretch it out for us so that it does get to this kind of stretched out state. That's the beginning of cytokinesis. After that, we are going to continue on through telophase, and really by the time we finish telophase, we're going to finish cytokinesis. So this is the timeline that we're looking at here. We start during anaphase, we finish during telophase. So this is a process that overlaps with the mitosis steps. When we talk about animal and plant cells and the differences with them in regard to cytokinesis, it really is all about the outer covering, because animal cells are surrounded by an extracellular matrix, and that extracellular matrix is flexible. But in contrast to that, the plant cell is surrounded by a cell wall, and the cell wall is very rigid. Okay, so this will change the way that we approach cytokinesis in these two different cell types. So if we talk first about the animal cell, and I draw the animal cell here, we are going to have that elongated cell, and it was elongated really during the anaphase process by the polar microtubules. Well, what's going to happen in the animal cell is that we will get a ring of proteins that begins to form around the center of the cell, or kind of around the waist of the cell. These are going to be a series of actin proteins. And what these actin proteins are going to do is they are going to slowly shrink. And when they shrink, they will start to pinch the cell. And so we will see that this cell turns into this cell, where we start to see that we have this little contraction down the middle. And this kind of indentation that we see here, this is what we tend to call a cleavage furrow. And this process will continue until ultimately we do get that cell pinched into two individual cells. And both of those would have a separate nucleus inside of them because the mitosis process took place as well. So that's cytokinesis in an animal cell. Overall, we refer to the whole process in animal cells as cleavage. And this is necessary to get those two independent daughter cells at the end of the whole mitotic phase. If we talk now about the differences and what's going to happen in the plant cell, in the plant cell, we have more of a boxy or rectangle cell. It's been stretched out, again, because of the polar microtubules, and we would have a separate nucleus on either side of this elongated cell. Since we're not able to just pinch the cell really down the middle because of that rigid cell wall, instead, the approach is going to be to build a cell wall down the center. To do that, we will have the Golgi apparatus is going to produce vesicles that contain cellulose. And cellulose is really the material that makes up the cell wall of plants. So we're going to have really little bubbles that start to form down the center of the cell. And then within these bubbles, if I just draw it in a different color here so to indicate that it's there, we're going to have our cellulose. Okay, so the green is representing the cellulose, which is the cell wall building material. So eventually, those vesicles are going to grow. So the vesicles grow. And when they do that, they will run into each other. And let's draw that here. So this is going to become a cell that looks like this. And now we've got a big vesicle really down the center where all of those have run into each other. And we still have this, really it's a cell wall material down the center. That is going to be what we call the cell plate. So the cell plate is the beginnings of the cell wall of the plant. And so here, once it continues to extend throughout, eventually we'll end up with the new cell wall built down the middle. Now, in addition to just getting the cell wall down the middle, it's also going to be lined with plasma membrane on either side, because each of these cells not only needs a cell wall around them, they also need that plasma membrane surrounding them. Okay, so that would be cytokinesis in the plant cell. That's going to take that one parent cell and it's going to divide it into the two daughter cells, which each has its own nucleus.